Hi everyone. Okay, so continuing with the review, getting ready for the AP exam, we're looking at some information about cells, which is part of Module 2. And so definitely wanted to look at a few things just to make sure that we're really clear as we're getting ready and studying for this exam. So the first thing I wanted to talk very briefly about was this concept of compartmentalization. So this is the idea that everything has its place. So rather than just kind of tossing everything in the middle and hoping for the best, everything is going to have a specific area. So probably the best way that I could give you an analogy for that would be the difference between an efficiency apartment and like a one-bedroom apartment. So an efficiency apartment is one where you walk in and it's got one great big room and then like an area over here with a stove and a microwave and a sink, right? And then a bathroom that actually does have a door. But everything else is this great big wide open space. There's no separate sleeping area. There's no separate like kitchen area as part of the living room, as part of the bedroom. It's a whole just one big space. So that would have less compartmentalization. When I lived in an efficiency, it was a little bit difficult to keep like work stuff out of the bedroom because it's just one big space, and so it's harder to keep it organized. In some ways it's less efficient just because you don't have like specific zones for stuff, so it can be a little bit harder, in that capacity. A one-bedroom apartment, on the other hand, would have like the living area, so it'd have the living room / kitchen area, a separate area for a bedroom, which would have a door, so there would be a separate space, and then it would have the bathroom with the door as well. And so the benefit is, visitors come over and they're not going to see your clothes out, they're not going to see an unmade bed, it's in a separate room. You can close the door, take a nap, and not look at the dishes in the sink. So it's just a little bit more organized. And normally, the bigger the living space, the more rooms it has. So some people will have a dining room specifically for eating, which they may or may not do in that room. Some people have a study, right, or a library, or a sewing room, a guest bedroom, separate bedrooms. And so theoretically, the more rooms you have, the more specialized those spaces can be. And so you know what goes in there and what doesn't. That's the concept. So as things get more compartmentalized, there's going to be some benefits to that. It's going to increase efficiency. So we know what's going to go where. We know what's going to happen where. Advanced functions. So if you have like a craft room, you can actually have like organization and things set up in that craft room, so you can have like ongoing complex projects. Like I don't have one right now, but I'd love to be able to have like my quilts ongoing, and then over here I've got my yarn for crochet, and over here I've got, I don't know, whatever else I'm working on. And you could have several different projects in different areas in a room rather than trying to put it all in a box and hide it in a closet or something, so it doesn't look so messy. And then the specialization, and it kind of all goes together. So the more separate spaces that we have, the more very specialized the individual parts of the cell and the individual cells can become. So when we're talking about specialization, as the level of organization found in that organism increases, specialization increases. So for example, if we were talking about the animal kingdom, sponges have cellular levels of organization. They have specialized cells, but they don't really have tissues or anything else. And so they're at the very bottom of that specialization, okay? And they're not going to be very advanced because they don't have the cells for it. They don't have any of that structure, as opposed to like an insect, which actually has systems. They're primitive, but they're still systems. So there's a level of reasoning that a sponge wouldn't have. They can do coordinated movement, sponge can't do, things like that. This is caused by gene regulation. So which genes are turned on and which genes are turned off during cell specialization from a stem cell is going to determine what the cell is capable of. So in my head, when I think about cell specialization, I always think of like that classic nerve cell picture, and then like, I don't know, a skin cell or a muscle cell. They look completely different. And it is going to allow some very advanced functioning. So even something like a primitive invertebrate, like an insect, has the ability to move. So there's a coordination and movement. Decision making is not normally something that we see in a lot of the invertebrates. We see more like hardwired or instinctive type movements. So the decision making would be more in the level of mammals. We normally see that. And so, for example, a chimp can pick up a rock and use it to break open a coconut to eat the fruit inside, or throw a rock to get fruit down out of the tree. So it's like concepts of using tools, working together, things like that. Increases with specialization. When we're talking about just the basic concepts of cellular function, here's what you need to be thinking about and looking for, okay? What kind of organism are we talking about? Is it prokaryotic? Is it eukaryotic? Is it a plant, an animal, a fungi? What are we talking about? Because that's going to give us a lot of information, okay? The next thing we want to think about is there evidence of flagella or cilia? If so, obviously, it's for movement, right? Because that's what the flagella and cilia do. Now, if it is, in some cases, that's going to be a cell that can move by itself inside a larger organism. In other cases, it's going to be like the cells lining your respiratory system that have little cilia, so that when you breathe in dirt and grime and things like that, because of pollution or whatever, you are able to, the cilia will help bring it back up, and you can cough up some of those things out of your lungs. So the cells themselves don't move, but the movement of the cilia is going to allow movement of something within the organism. So some sort of movement, right? Flagella could be anything from a bacteria that can free swim. It could be a protist, even a parasitic protist, like Giardia, for example. That would be an example of something that needs to swim in the body in the water. Or it could be something like sperm, which has to get from point A to point B in order to fertilize. So we're looking for something with movement. The other things that I want you to think about if we're looking at a eukaryotic cell is the amount and the presence of specific or specified organelles, okay? So if we're looking at it, and College Board makes an intentional effort to talk about a specific organelle, there's a chance, not guaranteed, because it's College Board, right? But there's a good chance that they're going to be using that information in some way. So think about, what do those specific organelles do? And then with that knowledge, what would a lot of those organelles allow a cell to do? So things like your ribosomes and your rough ER. If a cell has a lot of ribosomes and rough ER, then it most likely indicates that that cell is making a lot of not just proteins, but probably advanced folded proteins, right? Because that's kind of the purpose of the RER. So you've got these highly modified proteins, probably tertiary or quaternary structures. Most likely they're not all going to be for the cell itself to use. I would be thinking that most likely it's making these protein products and then sending them out. So it could be producing protein-based hormones. It could be a gland cell making hormones. It could be a cell making protein messengers of some other type to send out. It could be a white blood cell creating antibodies and sending them out. something along those lines. So if it talks about a lot of it, we start thinking about why would it need so much, right? Or thinking about what those proteins could do inside the cell. Like, most likely, if it's a muscle cell, they're going to have a lot of ribosomes and RER because they need a lot of proteins to be able to move. So we just kind of think about how that could relate. If we're talking about the smooth ER, we're going to be talking, of course, about lipid, specifically phospholipid generally, production, and or we're talking about filtration of toxins, right, or detoxification. So the organ that is primarily used for that would be the liver. So in the liver cells, we would expect to see a lot of smooth ER because it's there to remove toxins from the blood. For example, alcohol, okay? It could be something like ibuprofen, medications, too much of a specific vitamin, whatever your body doesn't need and it's considering a toxin will get kind of hopefully detoxified inside that liver. From an anatomy standpoint, I would expect to see more smooth ER in an individual that happens to be a habitual who drinks a lot of alcohol because their body's going to have to process more than an individual that does not. So that might be something we're looking at. The next one on there is Golgi. Well, so we're remembering that Golgi is going to package and transport. So it's going to package, modify, and ship. So you're going to see a lot of Golgi in cells that produce things to send out. So think gland cells that are making hormones, sending them out, or anything else really that's making something and sending it out, or cells that are receiving a lot of information from the outside. So there could be something like that where it's getting signals coming in and then it's doing something from there. Okay, so we're looking at that. The next one up is mitochondria. So please do not ever write the powerhouse of the cell. We've talked about this. That is the bane of the reader's existence. But it is going to convert glucose into ATP through hopefully aerobic, well, for the mitochondria it would be aerobic cellular respiration, right? It's going to go through Krebs cycle and then it's going to go through the electron transport chain. So when we're thinking about the mitochondria, we're going to be talking about cells that have to make a lot of ATP energy to survive. So these are going to be things like neurons, cardiac muscle cells, and things like that. Cells that are motile are going to have some as well because they're going to need the ATP. The next one we have on there is chloroplast. So when we talk about plants, from the time we're little, we're made to believe that every plant cell has lots and lots and lots of chloroplast, and that's not true. If it was, every part of the plant would be bright green, and they're not, right? So thinking about that, we need to think about what parts of the plant are normally green and exposed to light to do photosynthesis, and the parts that are not green and not exposed to light are not going to have chloroplast. So roots, woody stems are not going to be part of that process. So we just kind of think about what is that plant cell, and if it's a plant cell but it doesn't have a chloroplast, then it's probably something besides a leaf cell. The last one on there is lysosomes. So we remember that lysosomes, I always use the memory trick of Lysol, is the thing that's going to clean up and recycle things that the cell doesn't need anymore. So when we're thinking about lysosomes in animal cells, plants have their own version. It's not called a lysosome. So in animal cells, a lysosome is going to be a little enzyme-filled sac that is going to take down things that come into the cell, and it's going to break it down if it doesn't need it, and it's going to recycle it. It's going to take part in breaking down things inside the cell that it doesn't need anymore, and then of course it takes part in apoptosis, which is programmed cell death. So something with a lot of lysosomes is probably something that's going to be bringing in a lot of material from the environment. So thinking about like a white blood cell, for example, that takes in invaders of various sorts and then breaks them down inside their cells. We'll be looking at lysosomes. Digestive tissue pulls in material from the digestive tract or the alimentary canal, and then it breaks it down, right? And then it releases those byproducts for the body to use. So anything that's going to be bringing stuff in and breaking it down, you're going to have a lot of lysosomes. So I hope that this was helpful. Remember that AP is not so much about memorizing everything. It's more about understanding processes and overall concepts, which is why I kind of focused on those today. If you have questions, I'm only a text or an email away. Please let me know. I'd be happy to help. Wishing you all the very, very best of luck on your exam. I know you're going to do great. Mwah. See you soon.